This is Garth Ehrlich. I'm a professor of microbiology and immunology at Drexel University College of Medicine, where I also direct the Center for Genomic Sciences and the Center for Advanced Microbial Processing. Today I'm going to talk about the development of machine learning algorithms to data mine pangenomic characterizations of complex bacterial species. I'm going to start with a discussion of chronic bacterial infections, which I've been studying for about 25 years. And I was at the very happy nexus of clinical and microbiological events, which led us to understand in 1996 that it was likely that chronic bacterial infections were associated with bacteria adopting the biofilm mode of growth. And this is an image of bacterial biofilms growing on the middle ear mucosa of the chinchilla. Subsequently, we showed that biofilms do in fact cause chronic infections in humans. This is a Streptococcus pneumoniae biofilm growing on the middle ear mucosa of a child who has otitis media or middle ear disease that has failed numerous courses of antibiotics. So you might ask, what role does biofilm play in getting to machine learning? Well, biofilms are what we call a population level virulence factor. And a population level virulence factor is a disease associated mechanism that exists or occurs only at the population level, i.e. it is not the trait of an individual bacteria, but rather a trait that's associated with bacterial multicellularity. And most of these population level virulence factors are associated with chronicity or persistence, as opposed to acute virulence traits such as toxin production. So one of the reasons that we were interested in biofilms is because biofilms potentiate other population level virulence factors, including horizontal gene transfer. So the biofilm brings bacteria in close proximity to other bacteria. So here we have two different bacteria, and the biofilm then provides a stable environment for mating, which allows the passing of a genetic trait from one bacteria to another, forming essentially a third bacteria now. It also provides a matrix for maintaining DNA in close proximity to bacterial cells for competence and transformation, which is a second type of horizontal gene transfer that bacteria undergo. So once we understood that bacteria in a biofilm were using multiple population level virulence traits, and we knew that horizontal gene transfer was operational, this led to my development of what we call the distributed genome hypothesis about 20 years ago. And the distributed genome hypothesis states that chronic bacterial pathogens utilize a survival strategy wherein a majority of their genes are distributed among a population or a species and are not found in all members of that species. Thus, there exists a supragenome, other people later call it a pangenome, at the population level, which is far greater in size than the genome of any one organism. And we have a virulence corollary to the distributed genome hypothesis, which states that this distribution of genes among a population serves as a supervirulence factor or a population level virulence factor that provides for improved population survival through continual horizontal gene transfer mechanisms which provide for rapid adaptation to environmental conditions through the reassortment of genes as well as alleles. But what's important to talk about here is that we're talking about truly the reassortment of genes in most cases, and not just alleles. So on this next slide is a cartoon of how we view a population-based supergenome or pangenome. So if each of the individual blue ellipses, which has a green helix in it, represents a strain of bacteria, and the green helix represents the core genome, that is the genes that are common among all strains of that population or species, what we now see is that if we look at the entire genome of each individual strain within that species, that it has this set of distributed genes indicated by a different colored helix. And so you can see that all strains are made up of a core genome and a distributed genome or an accessory genome. And if one of these strains dies, the DNA goes back into the matrix of the biofilm and then can be picked up by one of the existing strains, forming yet another strain. And so this is the concept of this super virulence factor, that the host is constantly dealing with the evolution of new strains through horizontal gene transfer mechanisms. And this is supported by the fact that if you look within a microbial species, there was no genomic diversity. Each strain or isolate within a species had its own unique set of genes derived from this population-level supergenome. The extent of this obviously was not known, but it was known that there were some genic differences among strains. So the problem became, how did we characterize this genomic plasticity and determine relationships among strains of a species? So with these thoughts in mind, we developed a strategy to comparatively analyze the genomic characters of large numbers of strains of several pathogens. And what this really was the development of next generation sequencing. So initially we partnered with 454 Life Sciences as their original academic alpha tester, which allowed us then to be able to sequence large numbers of genomes of multiple bacterial species, which was something that was not feasible prior to the development of next-gen sequencing. And so we chose a number of different bacterial species to look at. Here you see Haemophilus influenza, Streptococcus pneumoniae, Staphylococcus aureus, Pseudomonas aeruginosa, and Gardnerella vaginalis. And we chose these as our original group because it included gram-positives, gram-negatives, gram-indeterminants, what we term professional pathogens, that is, they live nowhere except in the human body, such as Haemophilus influenza and Streptococcus pneumoniae, and then opportunistic pathogens such as Pseudomonas aeruginosa and Staphylococcus aureus. Because we wanted to know, when we were trying to test the distributed genome hypothesis, whether it was going to be universal or not. And so as we started sequencing individual genomes of Haemophilus influenza, we plotted the total number of genes on the top here, so that on with the blue diamonds. And we also plotted the number of genes that were core, that is, that were present in every strain of the species. And so what you can see is that the core genome very rapidly reaches an asymptote of around 1,400 genes, but the supergenome or pangenome just continues to increase in size. And so we were interested if we could actually model this. And so Justin Hoag, who was a mathematician in my group at the time, actually developed what he called the finite supergenome model. And this is a graphical representation for Haemophilus influenza here. And so you can see that in Haemophilus influenza, you'd expect to have about 3,000 genes when you've got 24 genomes based on his modeling. And so what we found was that as we increased the number of genes up to 24, his model was actually pretty much right on. We were right at 3,000 genes when we had sequenced 24 independent isolates. And so what was very interesting is when we looked at the genomes of each of these strains, we found really enormous differences in the size of the genomes. The smallest genome had only 1,688 genes, whereas the largest genome had 1,967 genes. So remember, this is the same species, but you've got one strain which has 300 more genes out of very small genomes than another strain does. We can also look at the number of unique genes that are in each strain, and with only a couple exceptions, every strain has a fairly large number of unique genes. So this one has 60 genes that none of the other strains have. This one has 31 that none of the other strains have. Similarly, we can look at the percent of non-core genes or distributed clusters in each genome, and we see it varies from 16 to 28% on an individual genome basis. So on average, what we found was that each strain differs at 18% of its loci, or 18% of its genes, with respect to every other strain. And this has very profound implications in terms of strain-specific differences with respect to disease phenotype, pathogenicity. So once you start realizing that you have this enormous diversity in gene content, you can actually start looking at parameters that no one ever thought to look at before. And so we developed a number of parameters, including the similarity score, which is the total number of gene clusters shared between a pair, the difference score, which is the total number of gene clusters not shared between a pair, unique share, that is the total number of gene clusters shared only within that pairing, and then a composite score, a comparison score, is obtained by subtracting the difference score from the similarity score. So on this next slide, I just draw your attention to the average difference. So if you're looking here at all possible strain pairs for Haemophilus influenza that we had sequenced at this point, on average, every strain pair differed from every strain differed from every other strain by an average of 378 gene possessions, which is truly remarkable when you see that the average size of the genome is somewhere around between 1,700 and 1,800 genes. And so even with just 24 genes sequenced, we can see that the core genome is smaller than the distributed genome. So the core genome makes up 47%, and the distributed genome makes up 53% of the pangenome or the supergenome. And of that distributed genome set, 17% are unique, that is, they're found in only one strain. We got very similar results when we looked at pneumococcus or Streptococcus pneumoniae. We see the core genome plateaus at about 1,500 genes, but the supergenome just continues to increase in size as we sequence more strains. So Justin used his finite supergenome model actually to predict how many strains we would need to sequence to get various coverage levels at various gene frequencies. So if we're looking only at non-rare genes, that is genes that are present in at least 10% of the strains, to get 90% coverage of the supergenome, we'd only have to sequence 11 strains. To get 95% coverage of non-rare genes, we'd have to sequence 17 strains. And to get 99% coverage of non-rare genes, we'd have to sequence 33 strains. But if we wanted to get just 90% coverage of all population frequencies that would include rare genes that are present at less than 10%, we have to sequence 142 strains. And then he also used his mathematical model to predict that the overall size of the supergenome was over 5,000 genes, even though no one strain has more than 18 or 1,900 genes. So in this case, the supergenome is 3 times the size of the core genome. So thus essentially providing proof of the distributed genome hypothesis, where we said the majority of genes would be distributed. And so, without going through in detail all of the bacterial species we looked at, we can now say that the distributed genome hypothesis holds for every bacterial species examined. All bacterial species possess a supergenome. And this is just tabular data for 24 different bacterial species. And you can see that independent of gram stain, low GC to high GC, large genome size or small genome size, that if we look just down here in the bottom, which is an average of across all species, that 22% of the genes in every strain of every species we look at are distributed. So we conclude finally that the distributed genome hypothesis holds for all bacterial examined. The core genomes in all species are smaller than the distributed genomes, with one exception, and that's tuberculosis. Thus, the supergenomes are modeled to be multiple times the size of the core genomes, and the observed genomic plasticity provides a plausible means to understand the phenotypic diversity among clinical strains of that species. So once we knew that there were large numbers of distributed genes and that they obviously played a role in virulence and pathogenicity, we wanted to characterize novel virulence determinants within the NTHI supergenome. And we thought that the way to do this would be using statistical genetics, because the vast majority of genes, even today, for any bacterial species, are either unannotated or underannotated, if you will. And so our strategy was to sequence a large number of NTHI strains, including commensal strains, that is, those that are not associated with disease, and virulent strains, those that were associated with disease, and then group the genes from all of these sequence strains into orthologous clusters, and then determine the completeness of the supergenome using our finite supergenome model, and then build from that a supergenome hybridization array, which would contain all of the distributed genes, and then perform gene phenotype statistical association studies using this species-specific supergenome chip. So essentially, we would interrogate large numbers of strains and then obtain sets of disease-related cogs. And so that's exactly what we did. We started with 24 sequenced genomes and blast annotated all the cogs to identify gene families, and then used that to build an array, and then screened over 200 Haemophilus influenza strains on the array. And what we found were that there were a large number of strains that contained what we call this SLR-like domains. And so if we look at the statistics of this, we just use Fisher's exact test, comparing the presence of this these SLR strains, these SLR genes, in virulent versus commensal strains, and got a p-value of about 0.02. And so this was the first example that you could use unbiased means, or in this case, statistical genetics, to identify a presumptive virulence gene just based on its differential possession within the species. And so then I had two graduate students who then worked to determine whether these VHISLR proteins that were predicted from this gene family actually played a role in Haemophilus influenza pathogenesis. And so one of the first things that they did was to knock out all 4 copies of this gene in a virulent strain of Haemophilus influenza that we call FAAAS, and replace it with a kanamycin cassette, as you can see in this slide here. And what we showed was that the knockout had greatly decreased fitness in vitro biofilms. So if on the y-axis here is competitive index, and on the x-axis is days following the start of the experiment. And so if the knockout was equal to the wild type, then you would expect to see a straight line at a competitive index of 1. And what we see here is that by day 2, the wild type has a great advantage over the knockout, or the knockout is essentially doing very, very poorly. And it continues to decrease in prevalence within the biofilm out to day 6. So this at least indicated, that under anaerobic conditions, because biofilms tend to be anaerobic, that this knockout was at a disadvantage. And so then we did similar experiments, except in vivo. So we used the chinchilla as a model for middle ear disease, or otitis media, and we put both the wild type and the knockout bacteria, mixed at a ratio of 1:1, into the chinchilla's middle ear, and then over time sampled and looked to determine whether there was a competitive index for the wild type versus the knockout. And what you can see, this is by animal, so we had 6 different animals here, and the competitive index again on the y-axis, just like we did in vitro biofilms, shows that for all of the specimens that we sampled, whether it was the actual tympanic membrane or the effusion that was in the ear, that there was a great competitive advantage for the wild type over the knockout in this animal model of otitis media, which has been validated as being a very representative of human disease. So again, more evidence now, in vivo evidence, that this gene, this VHISLR gene, is important in pathogenicity. So at this point, we could conclude that the wild type had a fitness advantage over the knockout in co-infection studies, and that we thought that it was associated actually with infection in the brain, because we could recover the wild type from the brain, but we could never recover the knockout from the brain. As I said, the wild type also had a fitness advantage in vitro biofilms, but not planktonic cultures. So all of this supported the concept that it was probably associated with being able to deal with anaerobiosis. We did a complementation experiment where we put 1 copy of the VHISLR back in, and it partially restored the wild type phenotype for local disease, but we still didn't see any in the brain. Remember, the parent has 4 copies of the disease. So we conclude that there's a copy number effect based on this, the difference between the single complement and the wild type. So what was the mechanism by which this gene or protein provided for increased survival in a competitive environment? So looking at related proteins in other bacteria, we suspected that it might allow for survival in macrophages. And so we did an in vitro macrophage uptake and survival experiment. And here you can see that's exactly what's going on. So the wild type strain, which is in the dark blue, enters and persists for at least 3 days in the macrophages, whereas in the knockout, represented by the red line, all of the knockout bacteria are dead inside the macrophages within 24 hours. And then we can show that the complement behaves like the wild type as well. Interestingly, if you take a strain of Haemophilus that does not have the VHISLR, which is this strain RD, you can see that it's also dies within 24 hours when uptaken by macrophages, as shown by the green line here. But if we knock in the VHISLR gene into this strain, then it behaves somewhat more like the virulent strain, with the bacteria persisting at least for 2 days. So we can conclude that the VHISLR plays a role in macrophage survival and in multiple strains, as well as affecting its ability to biofilm growth. And we also showed it provides enhanced serum resistance. So we've named this protein now the macrophage survival factor. And so I went through this demonstration of characterizing an unannotated protein because I want you to understand that not only can we use, this unbiased method, in this case a statistical method, to identify a candidate virulence gene, but when you test the candidate virulence gene, it actually plays a role in virulence. So as we have continued with our work in sequencing large numbers of bacteria within the Haemophilus influenza, two of the computer programmers, actually three of the computer programmers in my team and my chairman's team, have now been using machine learning technologies, which are statistical methods, but they're very deep statistical methods, if you will. And you're able to pull much more information out with these deep learning methods. You can with just using a simple Fisher's exact test or student's t-test, as we did with the initial studies. So just to reiterate, Haemophilus influenza, which is our general bacterial guinea pig, it is a naturally competent species. It has molecular machinery that allows it to uptake exogenous DNA from its environment, and then it can recombine that DNA into its genome. And so if you look at the phylogenetic relationships, they do not necessarily reflect phenotype because of horizontal gene transfer. So the image on the right-hand side here you can see what happens when you get horizontal gene transfer. You would not expect B to have the trait that it does if you're simply using phylogenetics, but it's picked the trait up by a horizontal gene transfer from C. And so again, by looking at gene possession versus some sort of identifiable phenotype, it is possible to identify genes that are associated with that phenotype. So now, instead of working with a couple hundred genes, we're now working with over 1,000 genomes, characterized both by the disease they cause and the tissue body site from which they were recovered. And so we have each strain's genes annotated, or at least grouped into cogs. Excuse me. And then what we did is, after we've done all the clustering, we decide to look at the distributed genes which are not rare, that is, they're present in 10% or more of all strains, and also not the ones that are near core. So basically, we're looking at distributed genes that are present at between 10 and 90% of strains. We're removing the extremes. And you can see down below that we have grouped them by location of isolation and by disease. And so by location of isolation, we've got muscle and cerebral spinal fluid and ear and eye, lower respiratory and upper respiratory, joint space and unknown. And then by disease, we have it by carriage, so these are commensal, by COPD, chronic obstructive pulmonary disease, conjunctivitis, meningitis, otitis media, and other. And so if you look at phylogenetic trees, and then try to put gene presence or absence on top of this in terms of looking at location of isolation or disease of isolation, you see it's just a complete scatter plot. And so it's it would be very, very difficult, using phylogeny to be able to pull this out. And so this is just a slide showing the filtering that we used to get rid of the bottom 10% and the top 10%. So we started with 4,207 genes, and we filter out the ones that are very rare, which is a large number, as you can see on the left-hand slide. And we sequence out remove the core genes, which is also a fairly large number. And so we're left with 769 genes as features, which we then used in 2 different machine learning algorithms as input. Each gene feature is a vector of either 1 or 0, representing its presence or absence in a particular strain. So we use 2 different methods. We use neural networks and random forests, and then use gene presence or absence to do training. And so the way it worked was we trained the algorithms on 80% of the strains, holding back 20% to then test. And we did 5-fold cross validations, repeated everything twice. All analyses were performed in R using the Carrot package. And so using the neural nets, we got better than you would expect just based on random, because we're looking at here 5 different disease or disease phenotypes. We've got carriage, COPD, conjunctivitis, meningitis, and otitis media. So anything over 20% is better than better than just random. But it wasn't extraordinary. We got a maximum of 45% or 46% using this neural network approach. Using a random forest approach, based on body site, we were able to get an accuracy of 56%, so that's almost 3 times better than just random guessing, and almost the same accuracy for disease type. By parameter tuning, however, of the random forest, where we looked at the number of layers of trees, if you will, that is how deep the forest is. We started with 98 and then saw that did the best over a very broad grouping, and so looked in the space around that from 88 to 108 and found that 88 did the best from that. And so we said, okay, so we're still kind of at the high end here. So we dropped down and looked at another group of trees from 68 to 88, which gave us a best of 81. This is for the body site. And we did an analogous type of tuning for disease and ended up with 47 layers of trees being the best for that. And so then if we take a look at the actual results we got following this parameter tuning, we see that it didn't make a huge difference for the body site. It went from 56 to 58%, but it really helped the disease. And so we are now at over 85% accuracy for the disease, calling the disease. And for some of the diseases like COPD, we're actually as high as 94% accurate. And again, for meningitis, 90% accurate. Our biggest issue was being able to distinguish carriage and otitis media. And this isn't that surprising because when you get otitis media, the strains in otitis media tend to overrun the nasopharynx, which is where we sample the carriage from. So we were pretty happy about this accuracy of this disease-tuned random forest model. And so then we actually used that to go back and look and see what the program was using as the most predictive classifiers. And this is where it got really pretty exciting for someone like me who's interested in things that we don't know anything about. So that's why I call it a window into the genomic dark matter. Because if you look at the table on the right, you will see that 4 of the top 5 genes encode hypothetical proteins. That means we have no idea what these genes are encoding. That means that it's very likely that there's huge amounts of new biology that we will learn by characterizing, the genes and proteins that this classifier has identified as being most important in determining what disease is the Haemophilus influenza is causing. We're also using neural nets in a different way to begin annotation of these random forest-defined classifier genes. And this is work that's being done by Will Dampier and my chairman, Brian Wigdahl's group, in collaboration with Josh Earl and my group, who was one of the people who developed the random forest methods. And so working with another middle ear pathogen or otopathogen called Moraxella catarrhalis, which is Josh's organism of study. They started with 13,692 hypothetical proteins from the Moraxella catarrhalis supergenome, and using a very restrictive cutoff of 0.8 as 80% probability that the neural net was going to get it right. They got at least one go gene ontology term for 1,920 of these hypothetical proteins, or 14%. And using a less restrictive cutoff of 50%, proposed 50% accuracy, they were able to get at least one go term assignment for twice as many genes, 28% of that total. So we now have multiple machine learning tools that we're using in tandem to both identify unannotated genes and then start to give us clues as to what the annotations of those genes might be. And I want to stress that this is all done just starting from pangenome or supragenome characterizations of large numbers of strains for which we have clinical provenance. So I want to thank several individuals, Fen Hu on the science side, who worked with me on the development of the distributed genome hypothesis and the concept of population-level virulence traits, and really developing next-gen sequencing. On the clinical side, I want to thank Chris Post, who was my clinical partner for 20 years, an ENT surgeon who brought the model of middle ear disease to us, and from which all the work I talked about today, from starting with the biofilms, emanated. And finally, I want to thank Bill Costerton, the father of biofilm biology, for inspiration and support. When I first developed the distributed genome hypothesis, I almost got laughed off the planet, but Bill was a big believer in it and told me to keep pursuing it. Next, I want to thank the team at ASRI, my previous institution, who developed a lot of the original tools that I talked about today. Justin Hogue is the mathematician who developed the Supragenome model. Josh Earl is still with me and is the one who's developing all the machine learning tools that we're doing here. Louisa Hiller, who did a lot of the pneumococcal work. Ben Gianto was one of the graduate students who characterized the MSF, the macrophage survival factor. Zada Med, who was my genomics lab manager, and several other people in the bioinformatics team. And then my current team at Drexel University, the lab team, actually do all the sequencing, and the bioinformatic team. Again, we see Josh Earl and Rachel Ehrlich, no relation to myself, are the ones who have been developing the random forest models, as well as Josh Mel and Archana Bhatt. So thank you. And I'll be happy if you email me questions to respond as I have time.